Welcome everyone. Tonight we are opening the first session of Inside the Circle, a gathering series under BioRevolution with a small and created group. No big stage, no performance, just honest conversation between people who are actually doing things. My name is Elif Damla Karakolcu and I am your host tonight. I write BioRevolution newsletter, and each issue I explain one biotechnological revolution from market, science and future perspectives. My mission is to make biotech accessible, understandable and investable. I believe the future of biotech is built on competition, cooperation and competition at the same time. And I spend my days creating spaces where scientists, startups and investors can actually meet each other. Tonight is one of those spaces. I work as a communications director at Capital Cell, which is the world's leading alternative investment platform for early stage biotech and life science companies. It was born right here in Barcelona in 2015 and since became one of the largest investors in biotech startups in Europe. The mission behind Capital Cell is something deeply I believe in. Make high impact investment in life sciences accessible to everyone, not just big funds or the privileged few. Anyone should be able to contribute to the next biotechnological revolution in human health. That spirit of openness and access is exactly why we are here tonight. Now I want to introduce the people in this room, because every person here was chosen with intention. We have Andrei Andrachka, biotech strategist and founder working with early stage companies in synthetic biology, biomanufacturing and bioinformatics, helping teams figure out where science can actually become a real product. Özge Özgüç with us. She is postdoctoral researcher at the Institute of Bioengineering of Catalonia, where she develops stem cell-based models to study how mechanical forces shape early human development. Ani Amar with us tonight. She is venture team leader at DR Ventures, supporting the next generation of deep tech companies. Aaron New with us tonight. He is founder and CEO of Sysgenica, building a platform that combines genome scale design and large DNA synthesis to solve some of the hardest bottlenecks in biomanufacturing. Kirill Popov with us tonight. He is climate venture scientist at Deep Science Venture and CTO of Climate, working on decarbonizing the built environment, quite literally figuring out how to keep our spaces alive. Marco Triccio with us tonight. And he is research scientist at DIOF Labs, exploring the frontier of artificial biology. Imi Gonchi with us tonight, and his founder is Pioneer, building an automated context layer for biotech R&D, making it easier for researchers to find what they actually need to move forward. Sibel Ağlamaz with us tonight. She is a community builder who has led multiple communities here in Barcelona. She is currently running the Turkish STEM community and building a Turkish business hub in the city. Iman El Amrani with us. She is a senior financial specialist and business planning partner at Sandoz, and she is the co-creator of Adamo, a new startup active in Morocco. And we also have two brilliant science communicators with us tonight. One of them is Daniel Lutz. He is a founder of Purple, a behavior-based communication and change strategist who helps people and organizations actually connect with their message. And Belen Ray with us tonight. She is a PhD in nanoscience, combining scientific analysis, positioning the strategy and professional video production to turn complex innovations into clear market stories. This is the room. I'm glad you are all here with us. Now our speaker, I have to tell you something personal before I introduce him. When I was a university student, I was just starting to discover entrepreneurship. Bioentrepreneurship was one of them and was not even known term back then. But I believe in it, so I organized Turkey's first bioentrepreneurship summit called BioPic, and my first speaker at that event was the same person who is here with us tonight. That was more than 7 years ago. Back then, he had already built Labredoor, Turkey's first public laboratory and independent research center. He was the first person in the world to bring a hacker house and the biohacker space together under one roof in Kadıköy in Istanbul. He built a community of independent researchers at a time when nobody else was doing that. Since then, he has kept building things. He is the co-founder of GEEN Biotechnology. He has worked in biodefense, collaborated with US Army and NATO, and he mentored hundreds of young genetic engineers. And now, after all of that, he is here speaking at BioRevolution's very first event. Tonight's conversation will move through some of the most important questions in independent science today. Please, Arda, welcome.
Thank you for the invitation. Thanks.
And I want to have some cheers more
cheers.
to start. Thank you for being with
Hey
us.
guys.
Thank you.
I'll just give a little introduction of like how I came here. It's been almost more than 15 years, I guess. Yeah, 2026, 15 years. So I started my role in this industry in 2011 as a junior researcher in a university lab working on nanotechnology. I was just learning how to do stuff in the lab. I was lucky because I found some guys, PhD students, who had some money for research. They were using the university lab and they gave me the chance to fail. They gave me the chance to fail in the lab by myself, which led me to learn what I knew about genetic engineering today. And it was mostly trial and error, lots of reading, sleepless nights in the lab. So I continued to tissue engineering 2013. Simultaneously with some genetic engineering, I joined a company of my friends, a startup company supported by the Turkish government, which was basically a competitor to Bolt Threads, if you know the company, the spider silk company. We were like a tiny little competitor. We were using a very similar process, some different host cells, maybe different expression systems. Sorry?
When was this?
Say again?
When?
This is 2013 and 14. AmSilk was there, Bolt Threads was working on it. We were working on basically Cruvermyces lactis and Pichia pastoris. Bolt Threads also, they also used Pichia pastoris. We actually met in a conference at some point where we exchanged some information. But soon after our company failed, 2014, we ran out of budget because we were running on like government budget, and we looked for some private investment. We were like a small group in Turkey. Bolt Threads was already manufacturing the silks. We were just able to we were able to synthesize the monomers. We were trying to figure out how to polymerize them, how to produce the silk. So we were actually behind. And we saw that we were not able to compete with them because they're like a Silicon Valley company. We were young, we didn't know what we're doing exactly in terms of like commercial sense, and we ended up switching to another field. We decided that okay, we know yeast cells now, we know how to express genes, we know what is lacking in yeast cell expression. So we came up with some new plasmids, some new gene expression systems. We tried to commercialize them as kits, and at the same time, CRISPR was picking up. So at that time, 2015, CRISPR was being adapted to new hosts. It was like getting recognized, and we realized that Pichia pastoris didn't have any CRISPR adaptation, so nobody adapted CRISPR. So we decided to take that challenge. We adapted the CRISPR system actually simultaneously with a research team in Austria, Vienna, I think, Vienna Technical University of Vienna. A research group used almost the same technology, same plasmids, same. We used ribozymes. They used ribozymes because there were problems with expressing the gRNA, whatever. So we ended up developing it at the same time, and soon after we realized these are not gonna get to the market as viable commercial products, and we need to do something that is either groundbreaking or maybe like we should start building something that is for 5 to 10 years later from now. And we started looking at what is the core problem of genetic engineering, like what we should tackle to cause a paradigm shift in synthetic biology. And we decided that the main problem is how we do genetic engineering. It's the complexity of cells. It's the manual process of engineering. It's the unpredictability. The models that we use in computers, the genetic circuits that we design, they simply don't work in the cells right away. We need to try thousands of times in parallel with different building blocks. And this is just too experimental to be a viable R&D branch. So we decided to tackle that by providing the industry a new host that is more is adapted better for genetic engineering in a quantifiable way. So whatever genetic circuit that we integrate into these cells, we wanted these cells to be more simple, have less noise, have less crosstalk between genes, has less dynamic parameters, so that we can model more accurately.
Can I ask a question?
Yeah.
Do you mean a little bit like, and I'm probably the least knowledgeable here on the biotech level, but do you mean a kind of, you wanted to develop the cellular equivalent of the white mouse? Or?
Yeah, exactly. And actually, this was not an original idea.
Was it?
The standard lab strains of the white mouse.
Yeah.
Yeah. And actually, this was an.
They're terrible for biology.
Exactly. I think Craig Venter recently passed away. He was one of the first persons to actually practically try to tackle that, and they came up with a synthetic cell, which is probably the most simplest cell that has ever lived on Earth, with around 500 genes, and that's all. And they made versions of it. The last one I think was called SYN3 3.0. They basically deleted every unnecessary gene possible, but they end up with a cell that is basically replicating only every 20 minutes, and it was just too slow and too inefficient to host any function, any engineered function on it. So some other teams tried to optimize the E. coli cells that they were working on, and we had a different approach. We said, okay, we're not gonna change the host. What we're gonna do is we're going to isolate the genetic language inside the bacteria. So we're gonna actually establish a virtual machine inside the cell, like if I go IT a bit. Basically, this means for molecular biologists an independent replication, independent transcription, and independent translation system, which would give us some independence from the host. And we started developing a 13-gene double-stranded DNA, which is able to replicate by itself, transcribe its own genes separately, completely separately from the cellular machinery, and hijacks some of the ribosomes of the cell and allocates them to our system. It was a masterpiece. We worked on it for 2.5 years, full-time at a startup company. We had funding. It ended up quite well. I went to the US and spoke with lots of colleagues in academia, including Drew Endy. He actually helped us a lot on this, and I actually presented to him the technology. And I was planning, as a startup company, I was planning to make this like a commercial product, maybe like go to Ginkgo Bioworks, some other companies, like, oh guys, like let's try our chassis for your projects. Like, would you like to try? That was my approach. But then Drew said, like, okay, this is a great academic work. However, there is only 2 to 3 companies in the world who could use this technology, who could even try this technology, and they're all here in around Silicon Valley, in San Francisco. So go talk to them. And
Sorry, which host organism was it?
we started with E. coli. So it's like a bacterial. It was easier to go with the bacterial stuff. By the way, we realized later on what we developed was probably the world's first synthetic viroid. It was basically turned out to be a virus, just without a capsid. It didn't have the shell, because that's what exactly viruses did. They
around 2019. I got sick going through this stuff. It's all coming together.
And we, at that point, we already burned like half of the money we had. And soon I realized this is not going to be a commercial product at all. We're not going to raise another round. And this is going to fail miserably. And I went back to Istanbul, where our labs were located. And we had to make a decision, because an enabling technology is not a viable commercial product. So we had to use that technology, or use those skills, for an actual viable commercial product, which turned out to be a pharmaceutical, a therapeutics technology. So we turned the company into therapeutics technology. We used those skills, not even the technology, we used those skills to develop an oncolytic virus. But our company was not set as a therapeutics company. It was not funded as a therapeutics company, and that led to a shortcoming in the finances of the company. So we slowly went bankrupt, and that's right before 2019. It's around 2018. I was still joining conferences all around the world, trying to catch up with the SynBio community. And right here I want to mention something. 2017, CB Insights released a market map of synthetic biology companies, 67 companies there. And I personally like it a lot, because GEEN was right next to Ginkgo Bioworks, if you know the company. But the problem with that market map is, today, when I look back at that market map, top 67 synthetic biology companies in the world, founded by the best academicians, like the most successful academicians in the world, most of it. We were like a rogue company, and there were like only maybe 10 to 15 rogue companies. And I will explain why there were rogue companies to start with. But the rest were like proper companies with like academic backup. Today, around 50, I'm speculating a little bit, but the number is close. Around 50% of all those companies went bankrupt. And that is a very, very critical indicator for us. I'll come back to this later, but basically it was the top of the synthetic biology hype. And from that point on, lots of companies failed. The reason being is not that the technology and the technical skills and the staff or the equipment was missing. It's not that. Companies were actually building these advanced technologies, and in the lab they were generating very good data. The problem was there was no market, there was no ecosystem. So it was, we had this illusion of we would be able to find as much as money we need if we are able to show data to generate thrill for investors and the community, you know. So we just focus on our work.
Can I ask, what were the premises this hype was based upon? Like, what was the narrative going on for, you know, this like collapse of 50% more or less?
So basically, what caused this was the free money in the system. After 2008, after the crisis, government flooded the system with new money. Those money ended up in banks, and these banks chose to fund big corporates. Big corporates couldn't find any other better use than to invest the money back in the system. And that money ended up in the hands of startups, basically, mostly. I don't say it was like a bad idea by itself, but it was a bad framework. We had the money back then, and we thought that like we're going to have a Series A, we're going to have a Series B, Series C. If we need $300 million to scale up this technology, that money is available to us. But what we didn't see back then was the monetary policy will change. The amount of liquidity, the amount of cash, the amount of, actually, most importantly, the risk appetite will be there for the next 5 to 10 years. That was our assumption, because all of us believed in it. At that time, we just didn't see that was an illusion. Soon after those negative interest rates and like high risk appetite venture capital start to disappear. Before they disappeared, they actually shifted focus. They did something smart for themselves. They shifted focus from high burn, high risk, high technical risk companies to low risk companies. And that's how I will come to the asymmetric success of synthetic biology companies back in 2018. If you look at the map now, that same map I told you about, you will see that the companies who survived are either pharmaceutical therapeutics companies, which already have a working framework for long years. They have a set up value chain, and it is, the game is like pretty rigid. Like the rules of the game is rigid. The money is self-sustainable mostly. It's predictable, and it's well established. But the other companies, like companies that engineered new strains of cells to use as, I don't know, probiotics, or like the yogurt company, some agricultural companies using synthetic biology as well. They just were spending too much, and they didn't have enough cash flow, basically.
I worked at Ginkgo as a senior engineer for 3 years. And
amazing.
So was there when they started to, well, I got fired in the second round of layoffs from Ginkgo. So they, like, the way that they explained it to us, well, in the way that they just like and I don't think it's a secret, it's just that they failed to find product market fit for the products. So when you brought up the pharma verticals, and you can earn just such enormous profits from a biologic. So this is in biomanufacturing, it's engineering a cell to produce like an antibody or something that you give to patients, and you charge an enormous premium for it. So the cost of the biomanufacturing part is really trivial compared to all the other costs and the profit you can pull in later. So there's a very clear product market fit for biologic pharma products. But Ginkgo's thing was like they were trying to go for things that you could get a lower margin on. Like they made this cannabinoid like candy that you can still buy in Canada. That product really works, but a lot of them like just weren't like the margins become really tight, right? Like the best like alternative to this is like the beer industry. The beer industry sells such an enormous volume of beer, or bread as well. They sell such an enormous volume that like the cost is very, very close to what they're selling, but the volume is just so huge that it's not such an issue for them. But these intermediate products that they were trying to make, like Bolt, like they're trying to compete with petrochemical clothing.
Yeah.
Right? I mean, it's like really, really difficult to pull it off and to get a strain that produces with a really high titer. And then there were like lots of things with the process, because when you're doing the engineering, the scale up into the fermenter is really very different. It's very costly to go from identifying a lead and then scaling up. So these were problems that we were having. Like, with Ginkgo.
Maybe that's how I remember you. Were you in Berlin, 2018?
No? I wasn't in Ginkgo.
Okay. Because I have a story. 2018, we're in Berlin. We're organizing I'm helping them organize a synthetic biology conference, and Ginkgo is invited. And we're like all thrilled. Everybody is like showing off their technologies. Lots of synthetic biology companies, some startups. We're pitching to investors at all, and it was time for Ginkgo. Ginkgo came up to the stage. They started presenting, and in an instant, everybody in the hall, our blood froze, because it was not a presentation. Ginkgo actually that day had a statement and basically said, we are going to monopolize synthetic biology research and development. Like they basically said, the way how we do research and development in synthetic biology is changing. We're automating it, and we're going to be the main provider of this infrastructure and this framework, and you are going to work through us. And at that time, I hated it. I hated the idea, because it's like, it's taking like all the freedom and all the possibilities that we were, excited about. But then I realized what they envisioned is not exactly bad, because actually right now, what would be the best thing to do is to start for a revolution, for a biotech revolution in synthetic biology, is that we actually need to change how we develop these technologies. Now with the availability of AI, advanced robotics, if we invest enough capital, we as private sector, we cannot. But if enough capital is invested, actually this might change. And Ginkgo was like a demonstration of it. The problem with Ginkgo, I think, you can...
I dropped a G wrong.
Yeah, please let's discuss on this, because I have some ideas. Ginkgo, it's like $1 billion company.
He's staying for the
It's very interesting, guys. Ginkgo is a strategic company for the US. Ginkgo is like, it's so important if it reaches its goals. It's so important as an enabling platform for SynBio.
Why? Why exactly are cloud?
Basically, it increases our throughput of research, by automating the manual labor and optimizing it. So if that vision works, we're basically gonna sit on a computer and program our experiments in parallel, in high throughput, without human risk, by the way. I'm more on the virology side, so one of the biggest problems with synthetic virology is there is always a risk for your personnel to get infected. If you don't follow the rules, if you fail to, you're actually like these, you cannot imagine, these brilliant minds, they're personally going into the lab, touching the virus with their hands right now, today. And these are like postdoc researchers, PhD students. These are like the most valuable locomotives of the field. And these people are risking their lives. And this is completely unnecessary because we should be like, it's not a very complicated job what we're doing. It's not more complicated than surgery. But it's still very manual. If you go to Boston University, if you go to UCSF, it's the same thing. We still use like the techniques of like 60 years ago. Maybe the equipment improved, but whatever. I think it's like a big enabler for speed. It will reduce the cost of trying things, and we still need to try things. It's still very experimental. So I think...
How would it reduce the cost of trying things?
Basically, the labor cost will be out, mostly. And we can, at the same amount of time, we could manage more experiments than we can manage in a conventional lab. So we can run things in parallel. We can actually like reiterate experiments autonomously. We can integrate AI as well to plan these experiments, how to most efficiently phase them and all. I'm not like an expert on it, but
the biggest value you see is not any particular biologically produced product or even platform for such product, but the pure operational protocol.
In the revolutional sense, yes. Like if we are, if we have a mission as the world of scientists and engineers to drastically increase our output in the shortest amount of time possible, we should be focused on enabling technologies, because that's like the biggest bottleneck now. I think, in my opinion. There are valuable low-hanging fruits in the field, and companies are like picking them, and some very successful companies turn them into commercial products today. But that's like, compared to what's possible, it's like a very, very tiny amount, just because it's like a costly and experimental process, if you ask me.
And so this operational protocol, like this new experimentation loop, the goal of that would be to synthesize new organisms that express relevant targets, right?
Yeah, exactly. And it could be also exploring new tools like receptors, like some new proteins, enzymes, like discovery. It could also include bioprospecting or like de novo engineering, like engineering new proteins from scratch. Anything that needs some experimentation after whatever modeling or simulation we can do in the computer.
Okay, but then isn't the biggest gap now not in that first part where you develop a strain, but rather what you mentioned is scaling it up to a large tank and then actually purifying the product?
Can you explain it to me again? You mean like, isn't the scaling up
maybe
like downstream?
I'm definitely oversimplifying this, but to me it seems that getting the organism, it's hard, but that's the easiest part. And the hardest part is actually making that organism work in the large tank with the proper aeration and mixing, and then definitely if you want to make it a continuous process, that's the next step, and then purifying the product out of the mixture.
You're actually right.
That is actually like a big problem.
So that is
scaling
the
up.
bottleneck more than the initial strain.
I do think we are limited by the initial part as well. If you have the tools, like if you have the genes, if you have the enzymes that you're going to integrate into the organism, and you know the pathway, you know everything. Yes, like if you're able to synthesize the cell, the synthesizing cell is the easy part. But selecting a strain also is a process and also can be automated. And I think the manufacturing and scaling up part is also can be automated. Like the synthetic biology research centers now, I don't know if Ginkgo has them, but probably they have at least like the small scale ones. But I know that China has. They integrate this part into the biofoundry as well. So it's not a separate phase from engineering the cell. It's part of it. It's like a chain of processes. And I do think we are actually quite limited with the proteins, enzymes we have. If you only think about metabolic engineering or expressing a single gene or protein, yes, it's easy to engineer the cell, but a genetic circuit is much more complicated. And it's you need to basically do like a combinatorial assembly. It's like thousands to 10,000s of trials, and selecting those strains is also like a problem.
I have an example too. So, like some of the most like cells are really good at making proteins. Like that's like the no-brainer for a product that you would get from a cell. And these are the most useful genetically engineered things, right? Like antibodies and things that are sold in pharma. But a lot of these projects tank because of problems in the downstream processing. So you can isolate the protein from the cells either by secreting it or breaking them open and then isolating them. But then downstream the cell begins to, the protein unfolds. It has weird glycosylation patterns, like various things in the cell. And so by engineering the cell upstream to favor the downstream manufacturing process, you can bring a lot of value. And that requires like a lot of tests to be made, as you were saying. It's one example of how there's still a lot of value that you can bring to the whole value chain by the initial strain engineering de-risking that you do with the
I mean, I will contribute to actually what you're mentioning. I have a background in pharma, not the scientific part of it, but now we see that after AlphaFold in 2020, like now virtually we can just 3D produce all the protein structure that can be done. And afterwards, I did a research recently. Now with the use of AI and synthetic data afterwards, you don't need to run the trials even in like thousands of patients or like how it's going to fold or unfold. Now versus let's say 10 years ago, we do the drug development in 30% less time, and we can develop 2.5 times more drugs versus like 10 years ago. So it's like, for me, it is like really, as Arda mentioned, it is like really we need to use the accelerated things. And this was my, contribution, but at the same time I have a question. If it is like an open question session as well, or shall we do it in the end?
Yeah, we're...
Okay. So I work a lot with rare diseases. I'm trying to help physicians to diagnose them. And at the same time now we're taking part in a research project with multi-omics data. So I think that is kind of the future, with the AI and machine learning capabilities. But I'm always skeptical because imagine like 95% of the rare diseases, they don't have a treatment yet. So if you find the gene targets, then you can just do your genetic engineering, how you would like to do. I'm not a scientist again. But what I see here is that like the scale, how we can implement it afterwards, I find it problematic. Pfizer has something, Novartis has something, XYZ, they have something, but this is like a huge process, just like to, yes, you engineer, but after you need to do a stem cell transplant, and you need to have like a critical mass of cells so that the body can produce regenerate the new system. So how do you think that it's going to evolve in the next 5 to 10 years, or how are you going to do it in a scale?
I cannot say predict how will it evolve, but coming back to my previous argument, I think automating the pipeline of developing new drugs or agents or targets, or testing them at least in vitro setup. The more we automate, because we have the tools today, because we have ripe enough AI model, machine learning models, we have extremely precise robotics, and it's actually some of these companies in the SynBio market map, they try to do it. I was about to make that point. So when I said asymmetric success of these SynBio companies, it was one, pharma therapeutics companies, and second, it was the bioinformatics companies and robotics companies. And I realized that the commercial success of these companies basically it was about the readiness of the core technology, and it was cheaper for them to implement their new technologies. Right after, though, the robotics companies failed. I do not think, however, they failed because of technical problems. They failed because of the state of the market. They simply didn't have enough customers to generate enough cash flow, and they needed immense amount of funding, and that amount of funding was not possible only through private investment. They also had to be invested by the government. So in the US, these tech hubs, these I would say infrastructures, were partly funded by private, partly funded by government, but they were structured as companies, corporations. So their target was profits. And I think it was a ridiculous idea for Ginkgo to go IPO. I don't know why they did it. And after they did it, they had to justify it, and they put themselves in such a bad position that they had to, I don't know if I'm allowed to say that, but it's public data. They had to. They had to fake their profits by incorporating a company in China and paying themselves. Like, that's at least what I saw in SEC reports. I don't know.
I really don't know about that.
Take a look around. Everyone has an accent.
No, but it could be wrong. But that's what I saw.
They spun off companies, and then those companies became their clients.
Exactly,
right?
and
it wasn't that they're literally like faking companies, but basically they created their own customers.
Exactly. But the interesting thing was, China was doing something completely different. 2017, 18, we're talking with Drew. Drew has the blueprints. Drew, Stanford Bioengineering, Drew Andy. He has the blueprints. And China is investing in the biggest synthetic biology infrastructure ever on the planet. I think it was from Shenzhen University of something.
I'm just saying. Yeah.
And they basically set up a full biofoundry from the start to the end. The biggest infrastructure ever, quite impressive. One of the guys, researchers, I was in Berlin with him. He invited me there. But I didn't know anything about China back then, and I was skeptical, and I didn't actually I don't know if Drew went there and see it, but I haven't seen it. But what I saw from the concept was amazing, and the good thing was, it was a government investment. So basically, government said, we're investing this as a strategic asset, which will drive innovation, and their expectation was not profits. They just wanted a leverage, and I think the way we failed was that, like, we were so focused on establishing companies which generates profits. It was simply the wrong framework to me. And today I think actually, if we want like a synthetic biology revolution, I'm not talking about like this incremental improvements that has giant effects on the market that could easily turn into products like in agriculture or food, but more advanced, complicated ones, which requires more money to burn to get to the market. So I basically think now is time for some government in the world to heavily invest in infrastructure, but not just the infrastructure. We have quite a number of educated people, PhDs, postdocs, actually more than we need, and more than the private industry can hire, and more than government hires. So I think there is like huge potential, huge reservoirs of talent that we can actually focus on producing output. They just need, and these people, the funny thing is, like, why do we have entrepreneurs, right? As an entrepreneur, we are in the profit, we're in the outcome, we're in the impact, right? We like the impact. But if you ask, maybe 85% of these people, these entrepreneurs, synthetic biology entrepreneurs, they're not in that business to be billionaires. They're basically in that because they're excited about it. We like the impact, we like the product we do, we're nerds, right? Basically, if we had some good offer from a government, which is just enough to contribute to a bigger project than we can do like by ourselves, we could actually increase the likelihood of coming up with innovative, maybe more innovative technologies, maybe not to use today, but to use 20 years later. To me, it sounds more like the space race. Synthetic biology, to me, sounds now more like space technology. It's not ready for privatization completely. Like, there are parts of it ready.
Wouldn't that just create a new bubble then? I mean, if you have all this, if you rely on a government to for 20 years, whatever, invest so much money, people would go into that industry, and at some point it's going to have to either become financially self-sustainable or collapse, right?
Yeah, it's exactly. That's why, actually, because it bankrupted our private industry who invested in early synthetic biology companies, because basically it was a ridiculous idea by itself, because the math didn't work. We didn't think it through well because we were too excited. Simply, governments can do that and get away with it. They can spend
unless they lose the election.
Exactly, and they'll be fine.
The thing that like Shenzhen had a really heavy governmental role in shaping their bio incubator hub. So what they have, from my understanding, is that they have like ginkgo style little startups that are making like genetic engineered organisms. But then they have like the rest of the value chain like, on the same campus.
Yes.
Yeah.
So like you can talk to people. There's less like, oh, I'm trying to find a partner, and you spend like a year trying to do that. Like it's a disaster. So it helps you validate your product really quickly and try to scale up faster by having that kind of government support is nice.
Yeah.
I think like there's a lot in synthetic biology that can be accomplished with technologies that were standard in 2005.
Yeah.
Right. With PCR and a little bit, even before
Exactly.
CRISPR is like you didn't really need
I mean, most successful bioproducts are actually not even synthetic biology.
Exactly.
This is like how pretty much any synthetic, any
You're
bioproducts on the
not
market is really just like find the right strain that's robust and makes a lot of something and scale it up and maybe modify it a little
That's
bit.
basically the low-hanging fruit, and there are plenty of them, and we can do that. That's like completely legit to do it. But if we want a leap in the industry, I think we need heavy government involvement. Also, there were private industry working in the space race as well. Like they were basically contracted by the government, which I think is a good model. We can contract research and development, and the companies will like speed the process up. They will be they will focus. You can like exploit your workers in these companies and like make them work 24/7.
Problem I see that China is maybe uniquely positioned, right? Both because of the political system and because of their economic interest to do something like this, right? I don't see Spain.
Oh, yeah.
Or
Yeah.
I mean, maybe the U.S.,
Yeah.
right? But like obviously the U.S. now has other priorities, so I don't know which other government could really. do something.
I heard.
European Union.
Of course they can do it. I think anybody can do it.
But like to do something like on such
they
large
have
scale.
they have tons of money to invest in this kind of stuff. I think like they're like they, you know, their priorities are. I think it would be better maybe as a European-wide.
In EIC, like Horizon projects and everything, they're like they're pretty good fun.
Maybe Europe collectively.
Yeah, Europe collectively. I mean, this is the
Under like a new
EIC.
maybe federal system or whatever that they can, or whatever. I'm not
In
speaking.
their latest fund, I think they released more than a couple of billion euros for like really biotech and health tech startups.
Biomanufacturing independence.
But the politics is a problem here.
Yeah, when it
We're-
comes for like each country, then every government is just for
Yeah.
themselves, and
Yeah.
5 years after, another one.
Yeah, I think they need to federalize. We're actually, it's funny that today we're actually on the fault line, the political fault line of the world in Spain.
But really, guys, like I don't know
Between
if
Europe,
you're
as a
familiar
Europe,
with the
yes.
funds, but EIC is like above the country. So
Yeah.
even you're from Turkey, we can apply for that. Likelihood is a little less, but you still can apply. Switzerland, anything, they're like pretty decent funds. Not at the level of
They're enormous.
US.
It's like way too much.
But and
It's like
they're
for a
doing
start- a
exactly
startup
the
doesn't
opposite
need
of what
4
we
million.
need, right? The fragment
A startup
stuff.
doesn't need 4 million or $1 million. A startup like
I need.
yeah. Well, you're in pharma. Yeah, you're in pharma. I don't know. Like
No, I'm not in pharma, but like I'm a
I mean,
startup.
I mean,
and-
like it could sort of intermediate scale funding, like non-diluted funding. That's like less competitive. EIC is really very competitive.
It's
So
very
it'd be
competitive.
competitive.
Yeah, exactly.
Yeah,
It's
yeah.
very
So
competitive.
something like for more mediocre ideas.
Yeah.
I mean,
Or like or just like with smaller packages for people to get going.
There are either other Horizons programs that can be applicable, like for smaller or other companies, like it's not only IC.
But what they're doing, what all this funding is exactly the opposite as like Shenzhen, right? Instead of building the whole thing in one place where everyone has access, they're doing the opposite. They fragment. They give all this money in small tickets to a variety. So their policy is exactly the opposite of what needs to be done, right?
Yeah.
Well, the thing is, there's already quite a bit of infrastructure in Europe in terms of the equipment, the research centers. Then the question is that if something happens there, it basically stays there forever. There's no way that's getting out as a reasonable spin out, because there is no standard rails to move IP out as a company. And with most of these institutions, it's always a bespoke, ad hoc negotiation whenever you want to do anything with them, even if you come from the outside as a paying customer, and of course even more so if it's something that they developed internally, right? And so I would say that most of the institutions, I'm not sure how it works in China, maybe you have more of an insight into that, but in the EU, most of the terms are borderline predatory. So
You mean for
VCs.
academic.
yeah,
academic
if you
research.
collaborate with an academic
Yeah,
institution,
yeah.
and
So
most of the
it's
infrastructure
operated.
is locked in the
Yeah.
academic institutions.
As an easy fix, I'm suggesting like having a shortcut and like giving up from our $1 billion company and IP, and let's just have a good paycheck and work for this vision together as employees. I'm personally down for that. As a like 15-year entrepreneur in synthetic biology.
So you'd say forfeit the IP from the start. Forget about it.
Yeah, like.
But then someone else will not forget about it.
Yeah.
No. I mean, like,
not as an investor, but there's some other party which comes after you a few years later.
Handing it over to the government as an academic, basically. Maybe with a good paycheck. Maybe with a good, I don't know,
Trust the
award.
government.
I do not trust the government a lot, but we don't have. I don't see other options working. I've been waiting and working on it for almost 10 years now, also working with governments on policy making and stuff. I don't see this happening fast enough.
Did we need to give the internet to the government? I mean, it was privatized, right? And so I think synthetic biology as a concept is just a failed concept in itself. So like if now we have to like make concessions for this concept to succeed, I don't see. I mean, I just don't agree. I think with that worldview.
Try this. I'm gonna flip it 180 degrees and say I'm actually an independent research enthusiast. I've been to many biohacker spaces all over the world, from BioCurious to Boss Lab to VAG in Amsterdam. And I've been watching closely what's happening in the world of citizens, regular citizens who are working in pharma, working in banks. And these people are enthusiastic people who would like to, who are just in there for the impact most of the times. And they just want some infrastructure to go into and develop projects, learn genetic engineering, whatever. And I found myself in a very, very interesting community with extreme potential. I realized that there are many more people than the people in academia who could actually contribute to scientific information and technological development. With that idea, I actually founded one of them in Istanbul in 2019 and opened this independent research center and accepted young genetic engineers, entrepreneurs from all around the world, and also gave them a space to live inside the place. So it was like both a hacker house and a biohacker space.
It was really cool. You were doing barbecue.
Yeah, it was fun.
Like this type of garden they had. So the scientists, I was super young scientist during that time, but we were going there, discuss something, and we were asking our questions to who is doing the PhD or graduates from the PhD. So it was super cool.
Yeah, and I've seen that liberation and that complete freedom, and without even money. These people are able to generate diagnostic kits. They're able to generate new materials that don't exist in the literature. These people can improve existing PCR techniques. There were some garage enthusiasts like working on droplet science who are developing these autonomous droplet-based, labs on plates. All sorts of different things, and I realized the potential, and the actual potential was these people are so enthusiastic about what they're doing. They continue whatever it takes, and they actually invest their own money, their own time, with no expectation, and they end up with the most efficient way of doing synbio R&D.
That is exactly my thing, right? So like now you're turning it kind of into academia where people are going to be exploited at the end of it. So like if people are just doing it out of their goodwill, eventually it's going to be expected that people do it out of their goodwill. So I mean, like a PhD is like slave labor to be honest.
Exactly. That's why I think this is a framework issue. If it is designed well, funded well, and inclusive enough, I think this potential can be leveraged and be used to produce immense amount of technological output. And I think this is a piece of, this is a way of competition for a government in a cold synthetic biology war. This has to be undertaken with a very high level of policy making and in government investment, I have to say.
But why? Like, why?
Also private, maybe, I don't know.
Why can't it succeed without all these? That's like my main.
Because we tried, and
So like we failed, right?
not all of us
Synthetic
failed.
biology.
No, yeah, none all of us failed. None all of us failed. These labs are still operating. I closed mine for actually partly personal reasons. But one problem in Istanbul was that the people we have didn't have the funding even enough to have like a hobby project. So they always looked up to frameworks where they can apply. So they went for iGEM, they went for some government support program subsidies, which are very small in size and take a lot of time. So it kills the vibe. You're still like waiting 1-2 years even to start a project. So I actually put my personal money in these projects, and like we just created 3 companies like that. But it's not enough for the whole potential. So we need a funding scheme, and I don't think private sector is good enough as a funding candidate, because they will expect profits as soon as possible.
So how are you going to sell this to a government if you are not selling profits? Because they are not nerds.
Exactly, because now they are in a war. They are in a competition, and the competition is fierce. And if you want to dominate, like Ginkgo wanted to do.
But what's the competition? What's the competition about? What do we create? That's like my main question. Is like, what has name one thing that synthetic biology has actually produced in this world? It's been around since like
golden rice, COVID vaccine.
Lots of things.
I would say that's genetic engineering.
GMO corn.
Yeah, the government just look at it
It's
for
the same
whole-
thing.
I would say synthetic biology.
It's the same thing. It's a fancy word for genetic engineering.
Yeah, it's genetic engineering basically. Yeah.
Arda, there is one thing I think that I suffered it a lot. In Turkey, we don't have even VCs who understand healthcare investment.
Yeah.
You know, they understand from GEEN.
Yeah.
Like there's a motorbike person. Okay, they're going to bring kebab from point A to C, and then I'm going to earn 30%. Okay, they understand this.
Yeah.
I mean, when you talk about synthetic biology or whatever, they just look like empty.
Yeah.
So there's one thing. Second, Europe is not much different, but there's more healthcare investment schemes. But that's also why, for example, I'm moving also my company to Boston, most probably, because like there you have access to private investments, you know, like you have top-notch institutions that you can support it, and I'm against government involvement.
That's
I
what
would
we have.
fire the half of the post office people that we have to use here in France, in Germany, and invest that money post office to science. It makes more sense. But do they do that? They don't. So don't expect anything from that mentality. I'm sorry, this is my take on the...
Basically, it's just that they can print money. That's it. They can print money. And that's the whole thing. Like, and they can print money for ridiculous reasons, and we just need a mental person to say, okay, go try. Because it's not logical. And these races are not logical. Was it logical to go to the moon?
Do you believe it?
But that's why.
I do. I genuinely.
Now there are like multiple wars.
Yeah,
now
probably.
We're fighting for investment versus AI companies and versus defense industry.
Yeah.
So even there are European calls about like, okay, how can we develop AI-guided missiles? You know, like, but they're not going to care about like, you know, life expectancy of a child. They don't give a shit.
Yeah. Like the other option is to develop an amazing like set of policies that will fuel the private industry with long-term incentives, like 20, 30 years in. So we're not worrying about short-term profits, or our investors are just secure. Then that could work as well. But our problem is like this short-term turnover times that are imposed on us, and that makes it almost impossible to have a leap. So we're only continuing to collecting the low-hanging fruits. But at some point, if you're collecting the low lemons from a tree, at some point you will need a ladder. They will like exhaust. Yeah, you'll exhaust those. I think at this point, governments, some governments, they need an edge, a competitive edge over the others. The globe is going highly polarized. The geopolitical tensions are rising, and I know from being in contact with these guys, I know that they actually like don't know much about each other, what they're doing, and they're afraid of each other, and it's an actual competition now. So this is like competition for world domination, and there are like new poles are generating in the south. Russia, China, US, Europe is now becoming its own thing. And they will have some sort of conflict, either military or technologically. I prefer a technological competition. So someone says, "Hi guys, like we have the best technology. Let's stop this," and like you submit.
Or maybe
that would be more peaceful, I think.
Maybe you could sell it as defense against climate change.
Exactly. That's one of the things that we need to prepare for, but not just climate. I would say there is increasing anthropological and natural biological pathogen risk, plus there is the risk of famine, there is the risk of clean water. Like we have global problems. These could be also centers for like main incentives for war or competition as well. Critical mines, raw materials, all these, like, so need for new processes maybe. There are many things that synthetic biology can actually solve in this thing, in this, and I would cap those under maybe biodefense, because biodefense is not just protecting the people from pathogens, but it's also preparing for natural challenges which are approaching.
Do you work in communications? How do you actually sell a? Because that's actually something that I'm very interested in. Is how do you communicate and sell a synthetic biology product? Because now you're talking about like the research of it, but like how do you actually sell one?
To?
Not just in general. Just
In general.
how do you sell a synthetic biology product?
It's going to sound really superficial because you're asking a super general question.
Yeah.
But it is.
Vertical that you would sell it in.
But it is. It's finding that really deep-seated human need and creating such a powerful story around it that people rally behind it and people say we want it, and then you get the momentum going. And once you have the momentum going, the other uses appear and people start coming on board, and you create an ecosystem. But it's the power of communication on the forefront is generating that momentum in the first place, because that's what's hard to generate. Once enough starts rolling, uses start appearing, partners start coming on board, and it starts becoming its own thing. So that's it.
Marco?
I mean, that's the actual problem of the whole thing we're discussing. There's no single coherent narrative that anybody can get. You can go to my grandpa, he's 90 years old, and he's gonna understand, oh, we're gonna make consciousness robots and stuff, because that's the AI vision, right? And you can sell that, even if we're not gonna achieve it. They have like a one sentence global vision everybody gets on board with. So, like my initial question like 1 hour ago was about the narrative. There was this hype, you know, the VCs or whatever, they had a bunch of money, they didn't know exactly where to put it, and then they chose, oh, let's put it in those synthetic biology companies, they look interesting. So it's, why did they choose that sector over, I don't know, let's make test here beer, or which is also synthetic biology, by the way. And so if there's like one, I mean, Heineken is doing it. They try to make like gluten-free beer taste like normal beer. Yeah, this is classified information. Anyway, you know, what's one sentence that like from your grandpa to some 5-year-old kid, they can agree, oh, this is cool, I want to do it.
Yeah, beer tastes better.
Yeah,
it depends on the application.
I think the impact was unmatched. That is possible with synthetic biology.
Yeah, but what is it? Like, I get it because I'm like a nerd, like everybody here, I guess. But what is it that you can sell to anybody that doesn't get it?
Well, for successful products, that's not an issue, right? Like if it is like a true 0 to 1, like you're curing a disease that you couldn't cure before, that has obvious value. You still need to do the work of selling that and getting that to market, but you don't need to wiggle your way there because it has obvious value, right?
Well, it has obvious value, but for instance, with neglected diseases, rare diseases, one of the reasons why most of them aren't solved is not because we couldn't, but because they don't affect enough people. So it's also, what are you finding that is close enough to home that the general person cares? Because it's never a technological issue only. It's getting enough people to...
I'm going to change it. I'm presenting the United Nations in July.
Good.
I'm going to change it.
No spoilers.
No
spoilers.
Do you work with the NDI at all, or no?
No, I'm presenting for AI for Good in Geneva. So it's like we're one of the top global 10 companies, AI for Good.
Nice.
And so the problem is that I'm going to do a little bit of a pitch, but like individually, yes, this is like little people, but approximately there are 800 million rare disease patients. 800 million. It's like the 3rd biggest country in the world after India and China. So if you look at it that way, yes, it is a big problem. And this is actually how I changed my pitch to make them understand the scale of the problem, because when you say rare, VCs, they run away. I mean, if it's rare, there is no potential, you know?
I'm not bidding for 3 people.
I mean, I think planes that are half the weight or concrete that can heal itself is impactful enough for like a, I don't know, investor to get excited and...
Oh, okay, I'm going to stop you there.
No,
that's what every founder thinks, and that's never the case.
First thing first, there is more alcohol if you need.
That's more than well.
Okay, please order what you want.
Synthesized on the moment.
Yeah.
That's
no GMO.
But that came out from
How many
iGen.
beers did you come? There were 5 beers, right?
I don't know, but yeah. Because when I joined the iGen, and there was this Dutch group, they were presenting an iGen product for beer. Now it's been a long time ago because it wasn't my bachelor's, but some time ago. But I'm like, more than 10. I don't remember.
Did you guys both do iGen?
I mean, iGen became like a factory for startups, synbio startups actually. It was like a good framework. It still is. Like 1 every 3 synbio founders was iGemmers at some point. It's an interesting, again, Drew's, I think that's Drew's project, is it?
He was one of the
founders,
The iGen.
Yeah. I mentioned him so many times today. Say hi, Drew. Like the last thing I want to mention on my side is that after this independent research project, I moved on to a biodefense company, which remained stealth to this date. And I was contacting governments, presenting them like militaries and all those stuff. Basically, I was trying to use the synthetic virology skills to prepare for the next pandemic and to provide the solution, potential solution, an option in a very bad COVID scenario, basically. We did lots of mathematical modeling, and we realized we were very lucky with COVID. And no vaccine technology actually could have saved, let's say, half of the world population. Because the mathematical modeling showed us with a virus as transmissible as COVID, which started as R naught 2 to 3, 3.6, so let's say 2.7. We had 60 days to save that 50% of the population. We had to discover, test, manufacture, and administer the vaccines within 60 days, which was, I guess it's impossible, was impossible with the technology available.
No, I think the phase 1 started already in April or May.
Yeah, but like
mRNA viruses. Right?
Yeah, but like to the point where we started administering them, it was like 1.5 years in, wasn't it?
Yeah, the end of the year.
Yeah.
No, it's 2021 got vaccinated. Yeah, maybe like
something around.
Less than a year
Yeah.
later, it was
Due to the clinical trials, guys, like normally, I mean, drug development lasts like 13 years with regular
We did a big
doses.
experiment.
And mRNA was not first used, but like in 2009 they were using it for prostate cancer. So somehow we got lucky.
Yeah, we got lucky. We got lucky with the virus. We got lucky with the vaccine, I would say, because the mortality rate was quite low. So there is something called virulence and transmission trade-off. So when a virus it says when a virus gets more virulent, so it kills more people by ratio, it actually has to trade off its transmission, so it's not that dangerous anymore. The problem with that idea is we now know that it's not always a fact, and until some certain threshold, increased mortality rate does not cause lower transmission, so does not decrease the R naught number. So we could have potentially like a 20% case fatality rate COVID, and it could be still as transmissive as the first strain. That was the risk, and maybe you've seen the news. There's this hantavirus now. It's an interesting subject. WHO today made like a speech about it, and they said like, no need to worry. I do not fully agree. I understand that it's not as transmissive as it could get. It's like a new adaptation. I'm expecting an R naught around 3 something. We're probably in the third phase of it, like the third generation or fourth generation patients are in now, and it's around 30% fatal. So case fatality rate is around 30 to 40%.
For each infected person?
So for every 10 person presenting with symptoms and gets diagnosed, 3 to 4 of them dies. Looks like within 5 to 6 days, which is good, actually. Technically, technically, technically.
That's why you're not in
yeah, because the longer it takes, and like the milder it is, it's harder for us to recognize it, and it could go and spread much easier. But I wouldn't be so optimistic either, because we see transmission. It's now appearing on different points in the world. It is still local because all these patients are connected with some contact, so it is under control. But I think it needs it needs to be responded right now. It is an outbreak. It's not a pandemic, but it has still the potential to go into a pandemic. Yeah. So I think we should react more seriously with these outbreaks. There is a good exercise for you if you want. There is a UC Davis project called spillover.global. In 2021, whatever in that list in the top 10, probably half of them to 60% of them had outbreaks in the last 6-7 years. So you can go online. It's very accurate, actually. I would say it's a very good project. I admire their work. You can go now, and like these viruses, they don't come from outer space. They're not like things that we haven't predicted. The prediction is right now online up there. If you go there, you can see in the coming 5 to 10 years what we're gonna experience. And there are some retrospective studies that said there is 5% increase in number of spillover events and 10% increase in fatality rate every year, which means I think it's like a very optimistic prediction. But in 2050, the spillover events will be 12 times more deadly and 4 times more frequent.
What is a spillover event?
Spillover event is when a new agent, new virus, let's say, jumps from an animal, a zoonotic or some other reservoir, into a human and makes disease. And most possibly, we recognize it when it spreads between, it transmits between human to human, with human to human contact, or like jumps from human to human. So basically, I even see the natural risk very significant, and we will at some point have another pandemic sooner or later. But I do think the anthropological risk is also increasing because our capabilities slowly but steadily is getting more mature. We were at some point playing with synthetic, semi-synthetic viruses.
You don't think we still are?
Huh?
You don't think we still are?
The point is, I would say, the pandemics and the agents that we're seeing right now are 95 to 99%, as much as like I can analyze, are natural. Or worst case scenario, it's an infected scientist while working with it, which is a bad scenario. But at that time in the world, there were like maybe 9, 10 more research groups who are capable as our team. And I think we cannot escape from that, the reality that this will be available to more people, to more governments. We don't have control over all of the governments in the world. We don't have control over all the groups or, infrastructures around the world. So someone eventually will come up with some technology that could exert force or pose risk. So I think the only solution to that is to come up with defensive technologies or technologies that can prevent harm. And I think that's like a very critical issue now. I'm still looking to work with governments at this point, I would say. But what I realized throughout these last 5 years was governments are very reactive in nature. I didn't expect that. I actually thought that these high-tech labs in governments, these advanced research groups and policymakers, they were like preparing for the next 20 years, 30 years. Especially in biotechnology and synthetic biology, we're actually doing very miserably. Like there are very good research groups and there are some amazing technologies being developed. However, we have no tools to protect the population from. Nice to meet you. Nice to meet you.
Nice to meet you. Thank you.
Yo, this is a small place, man. Alright.
And have you tried talking to the doctors?
Doctors? No,
The epidemiologists.
The epidemiologists. No, I haven't tried that.
Because they are who make the decisions.
Yeah.
It's like, for example, whatever you tell me makes all sense. Like future, they had DARPA, now in the US they have ARPA-H.
Yeah, ARPA-H, DARPA, yeah.
So we're talking to them as well, but like, I think the right English word is that with kind of pandemics, we have fear mongering. It makes sense.
Yeah.
So like COVID, I did this analysis like 2 years back.
Yeah.
During the time, it was like approximately 9 to 10 million died out of COVID, but at the same period, we had 25 million died globally from antimicrobial resistance.
Yeah.
So for me, it is like, there is like a
huge
big
risk.
event. I mean, by the way, I'm like pro-vaccine and anything, don't get me wrong, that it didn't exist. But there are like real life solutions as problems right now, and then we're not paying attention if we're preparing for like a pandemic in like 20 years, 30 years. So, I mean,
until it gets critical, the response I think is very slow. We should be start getting ready 20 years earlier to these kind of problems, because coming up with a solution takes 10 years at least. Gathering the people, making the research, clinically proving it or trying it, making sure that it's safe. These actually take time with the technology we have now. So governments has to be proactive about it. They are funding research through universities, I know, but in practice, today, no government has like a red button, like a B plan to protect the population from a pandemic or some superbug that is resistant to every antibiotic, right? And I think this is like one of the most important problems, as important as the subjects that were spending the most amount of money.
For example, E. coli is a pain in the ass. I mean, for
Even
clinicians.
the simple E. coli, yeah.
So
The most known like organism.
Or MRSA, I mean, actually,
The-
it's a little bit more complicated. But.
Thank you for this conversation. I will close the record, and after that it will be continue off the records, and everyone can put more idea maybe on the table.
And we can talk about it.
Yes!
Yeah, that's what we were saying. Actually, it didn't come out of the lab at all. It did come from outer space.
Thank you so much for being with me tonight.
Thank you for organizing.
Yeah, thanks.
To Biotechnological Revolution.
Yeah.